So, good afternoon, class. Today, in the subject Zoology 2, Animal Physiology, we are going to see how we can generate the action potential inside a neurons. So, basically, in last lecture, we already studied about the neurons and how their structure work and what are the basic function of neurons, a special part of the axon, function of dendrites, and how they are going to start the information transfer process. So that all process we already has been studied. Now we have to see for their information transformation process via action potential of memory. So in the very first introductory part, we know that the central nervous system has been made up of like 100 billions of the neurons. That's a huge number. That like 100 billions neurons are going to form our central nervous system. So that's why this is a very complicated system present in all the physiological action inside our body, and all from all this genetic makeup of that whole gene structure of a particular organism, that 40% of our human gene, they are the participated, or you can say their basic function is to produce these 100 billions neurons. So their formation degree is huge, and their quantity too, because they needed to be perform a very specific kind of function that is play a very major role in the development and the, you can say, progressive features of human body. We talk about two specific kinds of the cell which has been present under the vertebrate nervous system, that are the glial cell basically. So we are having these two kind of glial cell. One is the microglial and second one is a macroglial cell. So this microglia and macroglia, basically in the microglia, they are the scavenger cells, or you can say they resemble tissue of the macrophages and remove debris resulting from injury, infection, and diseases. So basically, not a very good cells for our body to develop, because they are removing the debris, and the result of that, basically, we are having the injuries, infection, and disease inside our body. So that's why what we are going to use for our nervous system are the macroglia cells. Now, here, if we see what are the important type of these macroglia cells. So basically, there are three kind of important macroglia cells. Third one is the astrocytes. So these cells, how we can divide them in a three broad spectra on the basis of their presence. Okay, at what position and what kind of nervous system they are present. On these basis, we basically divide them like oligodendrocytes and Schwann cells. Oligodendrocytes are the basic component cell of the axon under the central nervous system. While on another hand, the Schwann cells, they are the axon component of the peripheral nervous system. As in last lecture, we already studied about the central nervous system and peripheral nervous system. So basically, for their differentiation for the central nervous system and peripheral nervous system, these are the different kind of cells which are present inside their axon. So oligodendrocyte, you just have to remember present axon cells. When we are going to talk about Schwann cells, it will be present in the system of peripheral nervous system axons. Now the third one, astrocytes, they have another role to play. They are present in a gray matter and the white matter of the brain. So the astrocyte, they are found throughout the brain. Okay, so basically they are not having a specific location to be present, like our Schwann cells and oligodendrocytes. They are having a location in a specific kind of nervous system. But the astrocytes, they are just present throughout our brain. So here are two types of the astrocyte cells. First is the fibrous, and second one is the protoplasmic astrocyte. So these fibrous astrocytes, they contain many intermediate filaments, or you can say most of the time they are primarily found inside the white matter. So they find throughout the body of the brain, but the special appearance of there will be in the white matter. On the other hand, protoplasmic astrocytes, they are found primarily in the gray matter, and they are having a granular cytoplasm. So these are the cell, and if we talk about the cytoplasm, so the protoplasmic astrocytes, they are having the granular kind of cytoplasm. Both types send process to the blood vessels, where they include capillaries to form the tight junction, making up the blood-brain barrier. So these both the vessels, they induce the capillaries. So in the blood, they have to induce the capillaries to form the tight junction. So in between what, blood and the brain barrier. So they needed to be balance it out, like what and how much account of knowledge can be transferred from brain to blood. This whole contribution will goes to the astrocytes. Now there are these three diagrams which are presenting oligodendrocyte, neuron cell, and astrocyte. So you can see the oligodendrocytes. This is the structure of the axon, and as we are talking about the oligodendrocyte, so they must be present in the central nervous system axon. So they are present inside the central nervous system axon, and they are their key feature of that particular neuron. Second one is the Schwann cell. They are present into the between the nodes of Rana. Okay, here and Schwann cell presenting inside the internodal gaps of Rana, and they are the peripheral nervous system cell. Next, third one is here astrocytes. So astrocytes, they can be present inside the whole brain component, but you can see the fibrous astrocyte image is being given here. So they are having some granular structure inside them, which make them a little special. And you can say the fibrous astrocytes, they are present in the white matter. Now, what does happen when the excitation and conduction happen inside a neuron cell? So basically, nerve cells, they respond to electrical, chemical, or mechanical stimuli. So any kind of stimuli has to be responded by the nerve cell. It can be the electrical stimuli, can be the chemical one, and can be the mechanical stimuli. All these kind of stimuli can pick up the reflex action inside the human body. So two type of physiochemical disturbance are produced. So when these stimuli are touched or passes through these nerve cells, so there are be two kind of physiological disturbance can be happen. The very first, that is called the local non-propagated potential, called depending on their location, synaptic generator or electronic potential. The first one is that. Second one is propagated potential, the action potential or nerve impulse. So the very first is a synaptic. We will take it one by one when we are going to study further. So you just have to be remember that these two things are different pathway, or these are two kind of difference physiochemical disturbance which will be happen at the time of excitation and conduction. So for these all things, so you can say these all comments, if you wanted to study them, so you should be know the very first important thing, that is called the resting membrane potential. And this resting membrane potential, it is very important during the generation of action potential inside the neuron. How it is going to be important? The very first, because if resting membrane potential, we don't know the specific amount of resting membrane potential, we cannot be know that it what potential is there going to be information. So first you have to know what is resting membrane potential. In short, we can call them RMP. So this is a voltage charge difference across the cell membrane. So cell membrane have two regions. I'll show you the diagram. Okay. So there are two matrices which going to be present inside a neuron. One is the extracellular, okay, outward to the membrane, and second one is the intracellular, that what inwards to the neuron. So here, if the what will be the voltage difference between these two cells, that will be known as the membrane resting potential. Okay, so this you can see the extracellular matrix of the cell they're having the positive charges in the form of sodium just outside the membrane, and you can see they are having the negative charge towards the inside the membrane just opposite the positive charge. So you can easily see that the inner negative charge, this is less as comparison to the external positive charges. So external positive charges are more, internal negative charges are less. So the difference between this positive and negative charges, that will be called as a cell membrane resting potential. In neurons, the resting membrane potential is usually about minus 70 MV. So microvolt. So this is the potential which can be present inside the neurons, and this is very close to the equilibrium potential of the potassium. So because this whole potential thing that is so close to the potential, or you can say equilibrium potential of the potassium, that's why potential even can make channels when the membrane is at resting position. So, because there are more open potassium channels than sodium channels at rest, the membrane permeability to potassium is greater. So, that's why there are more potassium channels will be formed if we compare them to the sodium channels at the rest position, because their membrane potential will be almost same. That's why permeability to the potassium is greater if we compare with the sodium. Now, the resting membrane represent an equilibrium situation at which the driving force of the membrane permanent ions down their concentration gradient across the membrane is equal and opposite to the driving force of these ions down their electrical gradient. So now, if we talk about the resting membrane potential, how what represent, so they are going to represent an equilibrium situation. So how, which basically driving force for the membrane permanent ions down their concentration gradient. So you can see here on the basis of concentration, they can be go uh, outward or inward. You can see here these are the potassium that is present in a lot of situation inside if we compare them to the outside. So you can say the concentration of the potassium is high at the intracellular membrane as comparison to the sodium. So this is called the ion concentration gradient. So nitrogen concentration gradient, that is high outside, and this is called a potassium sodium ion concentration, that is you can say high at the inner side. Cl always high towards the outer side and downwards as a fluorine, that is downwards towards the inner side. So these are called the concentration gradient, and concentration gradient also have a very important role to play inside the action potential generation. So, a membrane potential result from separation of positive negative charges across the cell membrane. So, basically, this membrane potential, how they will be formed? They will be formed by the separation of positive and negative charges. So, this have to be balanced. Like you can say, the outer the membrane is the whole positive charge aligned via the membrane line, and the inner side the whole negative charge that can be aligned just inner side. So you can see that this have to be balanced to create the membrane potential. Now we will see the that was the membrane potential. Now we have to see what is action potential. So, a momentary change in electrical potential associated with the passage of an impulse along the membrane of a muscle cell or nerve cell. So, what happened? An action potential. This is a neuron way to transporting electrical signal from one cell to next. So if a neuron cell wanted to transfer a specific signal from one cell to another cell, then they needed to be generate a electrical potential. Okay, so this electrical potential, and it have to be associated with the passage of an impulse. Okay, a impulse, electrical potential, this both the thing, they have to be needed present outside of the cell matrix. So When this electrical gradient formed, there will be a ion channel formed inside the membrane, and along with the outer ions, some impulse can be come along with them inside the cell just to transfer their impulse or information. So this kind of mechanism, that opening of the ions channel with the help of electrical potential, with the impulse section, so all this process happen inside the neuron just to passes the information from one cell to the another cell. So I think you got what is the meaning of the action potential. If we go a little deeper in that, so the action potential are the primary electrical response of neurons and other excitable tissues. So they are main form of communication within nerve system. So basically, these action potential, what are they? They are primary electrical responses of neuron or other excitable tissue. If the muscle tissue there, so that in that case we are calling them the excitable tissue. So they are just the result of any excitement or electrical response. So what they are doing, they are just communicating within the nervous system. So within the nervous system, from one cell to another, that cell to another. So this whole communication process inside the nervous system, that basically formed over these action potential generation and conduction. So they are due to change in the conduction of ion across the cell membrane. So the cell event in neurons are rapid, being measured in milliseconds, and the potential changes are small, being measured in millivolts. So here, like you can learn it like some definition point of view, like the electrical events in neuron. So these electrical things which are going to generate the section potential, they are very rapid. They're not slow. That is a very rapid and fast process being measured in milliseconds. So you can say a unit that is very small, even from a second. So the millisecond, and the potential changes are very small. So that the potential changer, or you can say ion concentration, electrical gradient, concentration gradient. So whatever the potential will be going to change there, that will be also very small. So that's why this potential that can be measured under the millivolt. So potential will be measured in the unit of millivolt, and the time which have to be taken by the neurons, that is very rapid, that will be covered under the millisecond. So this is all the definition part. Now, how we can going to generate the action potential? So, a neuron that emit an action potential is often called as fire. So, the very first, if suppose a neuron get information from somewhere, the very first neuron, and if he wanted to transfer that information to the another neuron under the central nervous system, so that neuron which is going to transfer that information will be called as fire. Action potential are generated by special type of voltage-gated ion channels embedded in cell plasma membrane. So what are the key for the generation of action potential? They are the voltage-gated ion channels. So these voltage-gated ion channels, they will be going to generate the action potential. How? We will see it. The rapid influx of sodium ions cause the polarity of the plasma membrane to reverse, and the ion channels then rapidly inactivated. So basically, what happened in a very first stage for generating that, there will be a rapid influx of sodium ion. So rapid influx means sodium ion that are present towards the outer side, they will be going to inner side. So this basic phenomena of that can lead to the ion channels to rapidly inactivate. Now, the sodium channel activation moves in a wave-like fashion. So now, then these sodium channels activation, they will be do it moves in a wave-like fashion. So basically, this whole channels that will rapidly inactivated. So the sodium channels activation move will be like a wave in a membrane. So first, the ion channels that are being inactivated. Now, what channels will be formed inside the membrane? That are the sodium channels. So the action potential is propagated down the length of the neuron from its input source at the dendrites to the cell body, and then down the axon to the synaptic terminal. So basically, if we see the diagram here, this. So this is a diagram of a neuron. These are the dendrites. Very first, if some impulse going to come here and the dendrite are going to catch that. So after that catching of particular neuron, what they are going to do, the very first they are going to transfer them into the stoma, and after that transferring to the stoma they will be transferred into the axon. So from primary dendroids to this terminal dendrites of the axon or terminal point of the axon, in this way the action potential conducted and transfer an impulse. This whole process of the propagation of action potential, that is started from the input source at the dendrite, and that will be done by the axon synaptic terminals. How does stimulus trigger an action potential? So when we got a stimuli, so how this stimuli going to trigger the action potential? So let's see this. The stimulus trigger an action potential in the cell membrane of the nerve cell, and that action potential provide the stimulus for a neighboring segment of the cell membrane. So you can see this. I think it will be easily understandable by the graph. So these are the some voltages and this is some time. Okay. So if this will going like this straight line minus 70 MV, or you can say minus 70 millivoltage position, that is a normal resting state voltage or action potential or membrane resting potential, you can call it anything, that is a normal thing. But if air will be threshold in the energy or change in the ionic concentration or some electric impulse get passed on in that field, this will be, like you can see, they can be reached up to the plus 40 millivoltage. So this plus 40 millivoltage of that much impulses, when they will become into just, you can say, to 1 millisecond. So in this thing, that whole synapse of the energy, that is called the action potential. Or you can see the rapid changes in between a normal resting position and a impulse get electrical conduction from anywhere. So that will be known as the action potential. This I already explained you, like how potential spread as a wave of a depolarization. So you can see how there will be converting for every second of the time, or basically when the time travels, so the membrane potential that also has been traveled throughout the membrane. Now we have to see ionic flux during the action potential. So how ionic flux work during the action potential? So the conductance of the ion is a reciprocal to its electrical resistance in the membrane, and it is a measure of the membrane permeability to that ion. So what happened? This is a total opposite situation. Okay, if we talk about the conduction of an ion, so that is a just opposite to the electrical resistance. So here there will be a initiation of something, not resistance. So like what kind of thing will be initiate that particular formula? So in response of a depolarizing stimulus, some of the voltage gated sodium channel open and sodium enter the cell, and the membrane is brought to the threshold potential, and the voltage gated sodium channels overwhelmed with the potential and other channels. So basically what happened, there are numerous type of the channels will be going to form inside the membrane in response to a depolarizing stimulus. A depolarizing stimulus is going to come here, and that stimulus are basically helping the membranes to open the voltage based gates, or you can say the gate which are reliable on the voltage. So when we are getting a voltage, so the very first here, what will be going to open? The sodium channels. As the sodium channel is going to be open, then sodium enter inside the cell, and the membrane brought to the threshold potential. So also the potassium ions and other channels, for like chlorine channels and other channels or other ions which are present inside the cell membrane, they are also going to be activated and forming the way for another ions to come in or go out. You can see the diagram and graphs here. So change in the membrane potential and relative membrane permeability to sodium and potassium during an action potential. So in this whole diagram, this is the excitability, sodium and potassium channels. This can, you can say, at the zero, and when the time increase, or you can say the refractory period get along with it, so the sodium channels and potassium channels, they will be open and closed. So at the time of high excitability, both channel are closed. When a little time period after, or you can say in between the 0 and 1 milliseconds, the sodium channels are going to be open, and after that sodium channel close and potassium channel open. So this is the alternatively present ion channel action. So this first sodium open, then potassium open, then sodium channel reset and potassium channel remain open. So in the third wave, what happened, the sodium channel going to be reset their position, and the potassium channel remain open. So they will be the open. You can see the sodium channel, they was here, now they are resetting themselves. So resetting themselves means they have to be at their first time position. So, or you can say the very initial position. So in this manner, potassium will be intact, but the sodium channel going to be reset. Both channel closed in the high concentration again. So the when the excitability is high, both channels are going to be closed. So this is how they are going to be change in a membrane potential, that is relative membrane permeability to the sodium and potassium during an action potential. So you can easily see how a sodium and potassium ions go towards outer and inner side during this action potential changes. Now, feedback control in voltage gated ion channels in the membrane. So there will be two type of feedback control. One is a sodium channel exert positive feedback, and second one is potassium channel exert negative feedback. So here we are going to get a positive feedback from a sodium channel, and for that potassium channel exert a negative feedback. So both of their features together are going to control the feedback mechanism. So first here you can see this whole diagram. Depolarizing stimuli come here. Opening of voltage gated sodium channels, they will be open. Stop, how will they will be stop inactivation of sodium channels. So opening of voltage gated sodium channel and inactivation of sodium channels. So now the normal kind of the sodium channels, they will be going to stop. What going to be induced? Voltage gated sodium channels. So after that, there will be increased permeability inside the sodium, increased flow of the sodium into the cell. Now, depolarizing of membrane potential, again they are going to depolarize, that is called the positive feedback, that will be led to the opening of voltage gated sodium channel. So this whole process going on and on along with the stimuli they are getting from there. Now, what happened in the potassium channel? They are going to exert negative feedback here. So they are also getting the depolarizing of the membrane by sodium influx. So they are getting this depolarizing stimuli after having the sodium influx. So these sodium influx, they are going to open the gate for the voltage gated of the potassium channels. So this increase the intake of potassium inside the cell, out of the cell, increased flow of potassium out of the cell will be happen, repolarizing of membrane potential, and they will be start going for the opening of voltage gated potassium channel. So this is a repolarized method, they are going again inside the membrane. So this will be called as a negative feedback. So this negative feedback are just going to stop the action potential after some time. When enough amount of information that has been transferred or some other reasons, they will be going to stop after some time. So the all or none law is the principle that the strength by which a nerve or muscle fiber respond to a stimulus is independent to the strength of a stimulus. So basically this all or none law, that is a principal thing here. how the strength by which a nerve cell or fiber cell they are just have to be moved, or you can say they're going to be respond to stimuli just by this law. So if the stimulus exceeded threshold potential, the nerve or muscle fiber will give a complete response, otherwise there is no response. So whole this process that is only going to happen if the stimulus exceed the threshold potential. If there will be no threshold in the action potential, then there will be no response to a particular stimuli. So this is very important to getting a threshold inside the potential. So this is a threshold, this is a resting membrane potential, and these are the thresholds. The above minus 55 degrees Celsius, you are getting these are the threshold. So only these threshold will be able to get a response. Now here, electronic potential, local response, and firing level. These are also some terms which involve in transferring the information. So a non-propagated local potential resulting from a local change in ionic conduction. So basically, when a non-propagated local potential happens, they will be resulting in a form of normal ionic conduction. Although subthreshold stimuli do not produce an action potential, they do not have an effect of membrane potential. So if we talk about the subthreshold stimuli, so they're never going to produce any kind of action potential, and they are not having the membrane, or you can say they do not have any kind of effect on the membrane potential energy. This can be demonstrated by placing recording electrodes within a few millimeter of a stimuli electrode and applying subthreshold stimuli of fixed duration. So if you wanted to see it practically, so you can see that by placing recording electrodes within a few millimeters. So just put some electrodes within a gap of the few millimeters, and if you're going to pass a subthreshold stimuli, then you will be going to see that there will be no rising in the action potential. So application of such current leads to a localized depolarization potential change that rise sharply and decay exponentially within the time. So basically, they are just having one stimuli on that particular neuron, but they cannot be able to pass that information to another. So this is called like they are getting this self-hold. So this thing that is just happened for this session, okay, they cannot be take it for longer, or they cannot able to pass that to the another phase. So that's why we are calling them the membrane potential. Just they rise up here and drop here. Okay, they cannot be propagated like the normal threshold potential that made a single line, a straight line parallel to the resting membrane potential. But they are just having this straight line here, so they cannot be make that. Now, change in excitability during the electronic potential and action potential. So, what can be changes happen in the excitability when this electronic potential or action potential happen? So, if we talk about the action potential as well as during electronic potential and the local response, so threshold, the neuron, the stimuli changes. So, basically, in all the process, threshold of the neuron, there will be stimulation of the changes. So, without a threshold, there will be no stimulation, and if there will no stimulation, then there will be no information transfer, and if there will be no information transfer, then we are not going to any kind of feedback. Hyperpolarization response elevated the threshold and depolarizing potential lower it. So basically, if we wanted to see what is going to increase that, so the hyperpolarization that going to elevate the threshold and depolarization potential that go going to lower it. So as they move the membrane potential closer to the firing membrane. So this basically the firing level that are used inside the membrane for just to transfer the information. Now conduction of the action potential, how they will be going to conduct. So the nerve cell membrane is a polarized at rest with positive charges lined up along with the outside of the membrane, negative charge along the inside. So we already see that this nerve cell membrane, this will be polarized at the rest condition. How? That a positive charge will be present on the outer side and the negative charge present towards the inner side. So this whole positivity and negativity that basically resolve in a polarity. So every polarity is going to be abolished for a brief period, that is actually reversed. Positive charges from the membrane ahead of and behind the action potential flow into area of negativity represented by action potential. So when this action membrane potential happens, so their positive charges they have to be go ahead of the membrane. So most of the time they present the inside to the cell, like the potassium in the case, but when they are going to go outside, that is called the in the process of current sink. So by drawing off positive charges, this flow decreases the polarity of the membrane ahead of the action potential. So when the positivity going to be drawing off, so that's very obvious that their polarity will be affected, or you can say they will be decreased. So such electronic depolarization initiate a local response, and when a firing level is reached, a propagated response occurs that in turn electrotonically depolarize the membrane in front of it. So basically, all this happening of electronic depolarization that going to initiate the local response at the final level, or you can say that when the firing level is reached, they will be going to turn electronically and depolarize the membrane in front of it. So this depolarization basically a kick up in the action potential. The spatial distribution of the ion channels along the axon play a key role in the initiation and regulation of action potential. So this is very important that the in what way the ion channels are distributed and how the axon is playing their role. So you can say axon playing and ion channels, they are both very important for generating or regulation of the action potential. Voltage gated sodium channels, they are highly concentrated into the nodes of Rana and the initial segment in myelinated neurons. So if we see the how these sodium channels, they are at what place they are so highly concentrated. So they will be very highly concentrated at the nodes of Rana in the axon and initially segment in the myelinated neurons. So inside the initial segment of myelinated neurons and the ranivere, they are present abundantly. You can see also, like as I already talked about, this is a myelinated axon, and they are having this polarization inside the cell or outside the positive charges they are present outer side, and opposite negative charges present the inner side. So this type of mobilization, you can say they are called the polarization. But this kind of thing, like charges are going to be opposite to each other, they are going to make a channel. Or you can say the action potential increment can be done by these two things when charges get older. ECF, micelle, and oxygen, you can say these are the nodes. If they are like the this negative out and the positive inner, so they are the active node. Basically, they oppose their natural phenomena. In a natural phenomena, positive outside, negative inside, but actually they reverse it. Positive inner side or negative outer side. So this kind of phenomena that is called the active node, and if this is present like positive outer and negative inside, they will be inactive node. So this kind of phenomena is present inside this axon internodal formation. Now, orthodomic and intradomic conduction, they are also can conduct either in the direction of axon. So this, when the action potential initiated in the middle of the axon, so the two impulses traveling in opposite direction are set up by electronic depolarization of either side of the initial current sink. So in the natural situation, impulse pass in one direction only from synaptic junction or receptor along axon in the termination. So such conduction is called orthodromic conduction. So conduction is the opposite direction is called antidromic conduction. So what happened here? If there is stimulus, it will be present here, or it got it here. Or if they are going to the opposite direction, like to the receptor input side, they will be called as the antidromic. And if they are going to sensory output to the sensory part of brain to the another one, in a forward direction, that will be called as the orthodromic. Orthodromic in a layer, or in what condition they have to be go, that is called orthodromic. Antidromic, that will be opposite to the stimuli. So this whole process, they can be the you can say two depolarization basically cancel each other, and sometimes they cannot be further transform the information if the two or both of the depolarization cancel each other. So this type of situation also can be seen inside some stimuli. So this is very important, like how these stimuli are going to work inside them. And you have to be remember these somatic motor output from spinal cord or brain. So if we are talking about like this is the version where the information has to be sent, but they're coming from this angle, or you can say this is under the reflex section, they are going to stimulate that there have to be very much elaboration of that particular stimuli. So motor output or muscles output. So just remember that natural situation impulse pass in one direction only. So if the natural thing is going to be happen inside there, so only normal direction what happen, they just pass in a one direction, they cannot pass in the both the direction. So that's why if they are passing traveling in the opposite direction, or it can be set up by electronic depolarization, that can be called as a antidromic. Or if the conduction will be getting from the middle section but going in a forward direction in a good way, then that will be called as the orthodromic situation. So this is all about that how the action potential can be generated inside a neuron, how can potential generated, how it is different from the membrane resting potential, what are the cells which are going to involve in their formation, and what are the things which will be considered when the information has to be transferred. So this is all about that how we can transfer information by a nerve cell under our nervous system. Still, if you have any problems, you can go through these references. There are a very good explanation has been given under these reference section, and this third number, takes on initial segment and updated viewpoint. This is very, you can say, uh, elaborated version of how axon potential work, or you can say how they updated the information from one neuron to another neuron. Still, if you guys have any problem, you can ask me. I already updated your quiz on the elements. Just go there and give the quiz. That's all for today, guys.